Hello, my name is Ivan Topolsky. I work as a software developer at the Swiss Institute of Bioinformatics, and in this oral presentation, I'm going to show the work that has been done in our computational biology group at ETH Zurich on VPipe, our viral genomic pipeline as we currently applying it to SARS-CoV-2. RNA viruses have characteristics which have impact on the dynamics of their evolution. They have higher mutation rates, they have short generation times, and exist in a large population sizes. This gives rise to both inter-host and intra-host genetic diversity. Such viruses can exist as a mixture inside a host. Over time, new variants may arise which can carry characteristics that impact on the treatment of the patient. These might be resistance to a drug, or, as discussed in the literature about SARS-CoV-2, that might be a change of a receptor. Over time, such variant can be selected for and take over the viral population up until the point at which its impacts can be seen on the host. Traditional sequencing method can only show an overview. They cannot show the diversity which is happening inside the host. A solution to be able to see the diversity and see the rise of new variants much earlier would be using next-generation sequencing. NGS technologies allow for seeing such fine details, but they come with their own shortcomings. The reads are short and the method is prone to errors. To address these problems, a new software pipeline has been introduced by the SIB in 2017, VPipe. VPipe takes as input the raw reads coming from the sequencing machine and a reference for the virus being studied. It can perform quality control, it does align the reads, it generates consensus sequences, and then calls the SNV against this consensus. It can also generate haplotypes, either at the global level or at the local level inside Windows. The code of VPipe is available on GitHub. It now features a new installer for quick and simple installation, and the pipeline relies on SnakeMake. SnakeMake will help install the component of the pipeline automatically. Both our own components, which were developed here at the CBG of ETH, and the third-party component that we are using are all available under open source licenses, and can be obtained as packages on Bioconda repository. An example of such component that we have developed is Shura, which is in charge of reconstructing local haplotypes and calling the SNVs. It works by sorting the reads inside windows, then, per window, clustering similar reads together and eliminating error which will appear spread randomly all over and not clustering together. Once these local haplotypes have been generated, calling the SNVs with a high confidence and eliminating the error becomes easier. More information about VPipe and how to use it can be found on the Wiki. Since the beginning of our research on SARS-CoV-2, we have also released a tutorial to help other people apply VPipe on their own datasets. We have also introduced an installer. In a single command, it helps you to download the software and get it ready to be used. The tutorial has demonstrated it. To facilitate downstream analysis of the results, VPipe outputs its variants using standardized VCF files for the SNVs and FASTA files for the local haplotypes. Another feature is an interactive visualization report which helps people look at the various SNVs, look how they map to the annotated regions of the genome or other interesting regions, such as, for example, primers, and can be filtered by quality level. Planned future improvements include interacting with other SIB resources. For example, outbound links to the Swiss model database of the SIB will make possible to visualize positions of the variants on proteins in 3D. One of the current applications of VPipe is using it to help automate the sequencing of SARS-CoV-2 sequence. Swab tests, which has been detected positive by the Viollier laboratory in Switzerland, have been obtained by the Genomics Facility in Basel. There, sequencing analysis is performed using an Illumina MiSeq, and the raw data are uploaded into an OpenBIS repository. VPipe takes care of automatically downloading the raw reads from OpenBIS, performing the analysis, also doing quality checks and making reports to assess the quality of the results. The consensus sequences are then made available to the Computational Evolution Group for downstream analysis. The sequences are released through GSAID and also on the European Nucleotide Archive under project PRG-EB 38472. In the future, we also plan to release the raw reads on the Sequence Read Archive. This open release enables NextStrain to draw a phylogenetic tree of the sequences found in Switzerland. Currently, more than 1,000 out of all the 1,600 Swiss samples visible there have been processed by VPipe. This sequence have now joined the amount of information which is available from the Swiss National COVID Science Task Force to help getting a better picture of the evolution of the disease. This Swiss cohort has also been contributed to the collection of European sequences which were analyzed and published in Eurosurveillance. Out of the 713 sequences for Switzerland contributed to this publication, 681 had been processed with VPipe. In the coming weeks, the Computational Evolution Group of ETH will publish an article using phylogenetics tree to estimate the spread of the SARS-CoV-2 virus. Using the intra-host analysis possibility offered by VPipe, the Computational Biology Group of ETH will also publish an in-depth analysis of the genetic diversity of SARS-CoV-2 within patients. This publication will combine the Swiss COVID cohort and also nearly 4,000 deep sequencings available on the Sequence Read Archive. And with that, I would encourage you to come visit our website. You will find a mailing list where you can get subscribed to get information about new releases and other such announcements. You can find an email to which you can ask your questions. We'll find tutorials and much more information about VPipe. I want to thank all the developers on our team who have contributed to make VPipe. I also want to thank all the external collaborators with whom we had opportunity to work with and develop our current technology. Thanks also to all the past members who have helped too in developing the previous versions of the pipeline. Thank you for listening to this oral presentation.